right, it's 11 o'clock, so let's get started. Thanks for joining us today. My name is Carrie Phillips, and I'll be coordinating today's webinar. I'm here with Jackie Carville. Jackie's going to go over how to use DNASTAR's Lasergene Structural Biology Suite software to create and submit protein structure predictions. You may have noticed that your phone has been muted. However, we encourage you to ask questions. To do so, type it into the chat dialog box and send to host. I will then direct these questions to Jackie to be answered for the whole group. We are also going to be offering an attractive offer. Jackie will be discussing this at the end of the webinar. If you need any assistance or have any questions during the webinar, you can send a chat message to me using the chat box. And with that, I'll turn it over to Jackie.
Thanks a lot, Carrie. Let me just share my desktop with you guys here. All right, to get started, as a little bit about us, we are a global software company headquartered in Madison, Wisconsin. Our sales team, developers, and support network are all here on site, so if you have any questions, we have the full support staff here to answer you and use your feedback to make our software even better for the next release. So to go over what we'll be discussing today, I'll first be giving this presentation, including some background on DNASTAR as a company, our Lasergene Structural Biology Suite, which is our software package containing the applications you'll be seeing today, and some background information on our NovaFold application. Then I'll be getting into the software itself, giving a live demonstration, showing you how to set up and run these protein structure predictions, as well as some protein workflows within Protein 3D, which include working with surfaces, epitope prediction, and some structure alignment. Now, we were born out of the E. coli lab of Dr. Fred Blattner at the University of Wisconsin in 1984. We built a strong reputation during the Sanger sequencing years with our products that continues on today. While still offering some of these classic sequence assembly and analysis tools, we have expanded into the realms of next-gen sequencing, supporting all major next-gen sequencing platforms. In addition, we offer software to assist the work of structural biologists, as we're going to discuss more in this webinar today. Now, after all of our DNASTAR software has been developed for research use only and is not for use in diagnostic procedures. DNASTAR products have long been established on the desktop computer, but are additionally available on both the Linux cluster or the new DNASTAR cloud for flexibility in your purchasing. DNASTAR is the leader in citations in peer-reviewed journals. As you can see from this graph, for the past 29 years, more researchers have cited DNASTAR software in scientific journals than any other commercial sequence analysis software on the market today. All of these people using our software also have some great things to say about us. Here are a couple recent comments that we received through Twitter. You can get in touch with us quickly across all of our social media channels, which include Facebook, LinkedIn, Twitter, and YouTube, and let us know how DNASTAR is working for you. We'd love to hear from you. So our Evolution Suite is the first of our software suite packages, and this was designed with evolutionary biologists in mind and features tools for performing multiple and pairwise sequence alignments. This suite also allows you to assemble contigs of Sanger sequences and identify genes, along with capabilities such as virtual cloning and primer design. Our Genomics Suite provides all of the software necessary for next-gen sequence assembly and analysis. We support all major next-gen sequencing technologies and a variety of projects, including de novo or reference-guided genome assemblies, targeted resequencing, metagenome and population assemblies, large-scale multi-sample SNP analysis, RNA-seq, ChIP-seq, and microRNA alignment. Additionally, we also support workflows such as automated genome closure and cancer genomic analysis. And finally, our Structural Biology Suite provides tools for structural biologists to analyze protein sequences, visualize macromolecular structure, predict epitopes, and align structures. In addition, the Structural Biology Suite offers access to NovaFold, which is our protein structure prediction program. Now, this is the suite we'll be discussing for the rest of the presentation today. Now, our Lasergene Structural Biology Suite that I just showed includes our applications Protein, Protein 3D, access to NovaFold, as well as several other sequence editing and analysis tools. Now, DNASTAR began venturing into protein analysis software back in 1998 with the introduction of Protein. Our Protein application was created to mine information from protein sequences, helping to predict and display patterns, secondary structural characteristics, and physiochemical properties of protein sequences. In 2011, we introduced Protein 3D. Protein 3D is our application for exploring macromolecular structure, motion, and function. Within Protein 3D, you can use graphical views to visualize your protein structure, employ various prediction methods to evaluate secondary structure characteristics, and easily customize your structure rendering. In July 2013, we have introduced NovaFold. Now, NovaFold is our new protein structure prediction program. Integrated within Protein 3D, NovaFold features the rich graphical interface of Protein 3D, but with high-performance computing on the Amazon cloud. With this NovaFold introduction, we have shifted our historical licensing model. Now, traditionally, all DNASTAR software was purchased as a perpetual license on your desktop computer. However, with the introduction of the DNASTAR Cloud in our Lasergene 11 release last year, and the introduction of NovaFold here, we now offer more flexible licensing options customizable to your particular research project. So with NovaFold, you now have the option to set up annual licenses or licenses by month, week, or individual NovaFold structure prediction. Our sales representatives are happy to talk you through your specific research project and set up a licensing plan that's best customized for your particular needs. Now, this NovaFold application was born out of an ongoing collaboration with Dr. Zhang of the University of Michigan. NovaFold utilizes the Zhang Lab's iTaser algorithm to build accurate, full 3D atomic models of proteins with previously unknown structures. So why this iTaser algorithm? Why we chose to base our NovaFold application off the iTaser algorithms stems from the Critical Assessment of Structure Prediction, or CASP competition. This competition is a biennial worldwide competition where hundreds of teams test their prediction tools against newly solved structures. There is a blind study of over 100 structure targets, and prediction results are assessed by an independent panel. The iTaser algorithm has won this competition for the past 8 years. So if we just look at the numbers for known sequence entries in GenBank and then compare these with the number of known protein structures in PDB, we can clearly see a huge discrepancy here. Protein structure prediction programs are so important because they allow us to work with proteins that cannot be or have not yet been crystallized. So we can try working our way to close this gap. There are three major structure prediction methods I'd like to briefly talk about today, which are homology modeling, threading or fold recognition, and AbInitio folding. Now, the homology modeling method is the easiest case of all three methods here and uses a solved homologous structure for calculations. This method is usually successful for about a third of all proteins. Now, the threading or fold recognition method is a method that's often used for harder problems. This requires a similar structural fold to the predicted protein. Now, this method is usually successful about 50% of the time for proteins. And finally, AbInitio folding is for the most difficult prediction cases, which require intensive calculations on the biophysical principles of the protein. Because of this, AbInitio folding works best for small proteins, about 100 residues in length. Now, these two images that you're looking at in this screen demonstrate the accuracy of the AbInitio method. The image on the left was created using the AbInitio folding structural prediction method in the iTaser algorithm during one of the CASP competitions, while the image on the right was created using traditional X-ray crystallography. And in looking at these, we can really see how close these two structures are. So NovaFold uses a combination of both threading and AbInitio methods. The NovaFold method finds structure fragments from multiple templates, builds fragments not matching the template, and then further refines the model. Now, because of the large computational power that this method necessitates, NovaFold uses the Amazon cloud. First, your query sequence is submitted on your desktop computer with the Protean 3D application. With your Amazon Web Services credentials, the sequence is then sent to a cluster of computers on the Amazon cloud, where the actual structure prediction computations take place. When complete, the predicted models will be returned to the desktop computer and Protean 3D for your analysis. So why do we use the cloud for this particular NovaFold application? Well, the computational power that is necessary to run one of these structure predictions is too great for any normal desktop computer to handle. And what's happening is that millions of small computations are taking place in order to build the final model. And by using the cloud to facilitate those computations, many can be done at once and your predictions can be returned much quicker and more accurately. What is nice about this approach is that you still get the user experience of a desktop environment with NovaFold housed within Protean 3D, while all of this computational power remains in the cloud environment. So to wrap up, how does this NovaFold application compare to iTaser? Now, right now, the results you get from the two approaches are very similar. However, with NovaFold, you do get our user-friendly interface, dedicated personal computer resources, so you don't have to wait to submit your prediction into a pipeline, the ability to run multiple predictions at once, and our strong support staff to answer any questions you have. In the future, we're going to continue to improve this code with the collaboration of Zhang. Over time, DNASTAR hopes to improve the accuracy of the program even further, as well as Diverge to provide you with additional structure prediction analysis options. So I'm going to get out of the presentation here and jump right into our software. So the first thing I want to pull up here is our DNASTAR Navigator. And in looking at the Navigator here, this is our central hub, which shows you all of the DNASTAR applications that are available. What's nice about the Navigator is if you hover over any of these individual applications, you can see a nice pop-up menu that gives you many different support options for that particular application. This has links to videos, tutorials, and demo data that you can download if you're working with a DNASTAR free trial or if you're just learning how to use your DNASTAR software. Another really nice thing I want to point out about the DNASTAR Navigator is its integrated help. And so if I open up the help here from the Navigator, it allows you to do searches across all of the DNASTAR applications, which is really nice. And finally, from the DNASTAR Navigator, you have the option to launch any of these applications directly. From there, I'm going to go ahead and I'm going to open up Protean 3D and then walk you through some of the different features of this particular interface. After we're familiarized with Protean 3D, we can then dive into NovaFold. So to get started in Protean 3D, first we want to be able to download any PDB structure from the RSCB protein databank. So in our web browser here, we can simply go to the PDB homepage. We'll search for a protein. I'll search for this 7-TIM protein here. And if we want to download this, we can go ahead and download our files really easily from the PDB website. So I've already downloaded this file, and I'm going to go ahead and open that up in Protean 3D. So what I'll do is select File, Open, and then select my 7-TIM structure file here. So now we're looking at our 3D protein structure here within the structure view. I'll expand that up a little bit. And if we look at the top of the interface here, we can see several different options to look at our structure. So we can use the zoom button to zoom in and out of our structure. We can use the move tool here to move our structure anywhere around the view that we desire. And finally, we have two different rotation tools to rotate your protein structure around different axes. Finally, if we want to look at any specific residue within the model, we can go ahead and just click on that particular residue here in the structure view. And this pulls it up on the side here. And if I click off of that, I can really easily return our protein to its original position by clicking the Reset Structure View button. So we can also very quickly and easily change the style of our protein as displayed here in Protein 3D. So if you look to the bottom right of our interface here, we can see that there is a Style panel that I'll go ahead and open up. And after opening this panel, we can see many different tabs available for customization. So if we expand the Rendering tab, for example, we can see all the options to change how our structure appears. So maybe I want to look at this in a ball and stick form, or maybe I want to look at all the side chains available in the protein here. So we have many, many different options to adjust not only the rendering, we can also change the color of the structure. This is some editing in the sequence view we'll discuss soon, as well as even the lighting of the structure that we're looking at here. So this really, really gives you endless customization for your protein structures. Now just below the structure view, I want to go back over to the left of the screen and I'll expand up our sequence view that I want to talk about. Now here we can make selections at various points of the sequence, which will correspondingly be highlighted in the structure view. So if I select any of these green helical elements here, we can see that this region is then highlighted in the structure view. If I select any of these red tube icons, then all of the residues involved in a particular binding site will be selected along with the binding site itself. So I'll close that back down. And next we want to look at the Explorer panel up here in the view. I'll expand that down so we can check that out a little more closely. This view here allows you the option to select different structural elements of the protein. So if we're looking under the Molecules tab here, we can click on any subunit of interest, let's say it's A, and we can see this selection reflected in both the structure and sequence views that we looked at earlier. Also, if we uncheck the box for the A subunit, this disappears entirely from both views. This complete integration between all views and panel selections is one of the great things about the Protein 3D interface. One of the other extremely important panels in the Protein 3D interface is the Details panel. So I'm going to pull that back up here. This is down back in the bottom right by the Style panel. And within this panel is detailed information about your structure or sequence. So right now I'm looking at details on the entire structure as a whole, but again I can also look at specific regions. I can even look at specific residues and get details about those. So this gives us a nice option to really look at closely many different components of our protein structure. So one last view I'd like to discuss here before going on is our Analysis view. And I'll open that up, the tabs right next to the Structure View tab here. And this view allows you to apply a variety of prediction methods to the protein sequence. So the way you manipulate this Analysis view is through our Methods panel over here on the left-hand side. And I'll open that up and expand that down. Now in this Methods panel, you can see a list of all the available methods and check those that you would like to apply to the Analysis view. You can expand each method's synopsis within the Analysis view for more detailed information that's available. We're looking right now at some hydrophobicity plots that are available. And in addition, under the Methods tab, you can also have the option to look at some specific parameters for some of these different methods that you can apply, so you can really customize how those methods are applied to your protein. Finally, as I mentioned briefly earlier, as with the structure view, we also have some different styling options for this Analysis view as well. So we can change any of these colors within the Analysis view as we desire. The last thing I want to point out, as with all the panels that we've discussed today, Protein 3D has different document and rearrangement capabilities. So I can choose to detach any of these views to be able to move them around and really customize our interface. In addition, if I want to move, I can move any of these panels to different areas as well. So this just really gives you a nice way to customize your interface for your specific use. So now I'm going to go ahead, after we've familiarized ourselves with the Protein 3D application as a whole, we're going to go ahead and talk about how NovaFold, our protein structure prediction program, fits into this Protein 3D interface. So there are many different ways to access NovaFold here. The first, I can go View NovaFold, get it there. I can do File, Predict Structures with NovaFold. I can go under Structure, Predict Structures, and select it here. And finally, there is an option available within the Details panel that I can access NovaFold from there as well. I already have NovaFold opened up, so I'm going to expand this up a little bit. I'm actually going to get rid of our 7-TIM structure we were working with here so we can look at this a little more clearly. And what we're looking at right now is the preview mode of NovaFold. And this preview mode is enabled for users that are not currently logged into their DNASTAR accounts via the NovaFold panel. Now this mode goes ahead and shows you some pre-completed structure predictions, which allows you to view the results, analyze those, and gives you an idea of what you will see as output from running your own predictions. As said before, with your purchase of the Lasergene Structural Biology Suite, NovaFold, predictions do not come included. However, you will be able to access this preview mode, so you can play around with these pre-done predictions and determine whether NovaFold is something you would like to add on to your purchase. So I'm going to go ahead and log into NovaFold using a DNASTAR account here, and we can check out some predictions that I've already done. So now that I've logged into my account, I can see this nice message from NovaFold up in the right-hand corner, which tells me about my license that I have set up. Now this particular account has 2 NovaFold licenses that expire on this date here. That's if I decided to purchase by a longer licensing option. This might have been an annual purchase of NovaFold here. But in addition, if you decide to pick a different flexible licensing option where you've purchased by prediction, for example, you'll see the number of predictions that you have purchased reflected up in this upper right-hand corner, and that will update automatically as you start running those predictions. So again, just wanted to mention that you have a flexible licensing option where you can purchase based on how many predictions you need, or purchase unlimited licenses for a specific period of time. So now I'm going to go ahead and look at the toolbar over here in the upper left corner of NovaFold. And this is where you have all the basic icons needed to manage your predictions. So from left to right, we have the option to create a prediction here. This allows you to select your sequence files. For each sequence file you select, an unsubmitted prediction will be created within the prediction list. This is your run prediction icon. This officially starts running your prediction. The terminate prediction icon, which stops running your prediction and ends the use of all your cloud resources, and the option to delete a prediction. So this command will both terminate your prediction and delete any of your results data. So any of the action of these commands in the toolbar will be reflected down below in this prediction list. Now the prediction table summarizes all of the predictions associated with your account. This is really easily able to be sorted. We can sort by number of residues, we can sort by time submitted, or the specific molecule name. So I'm going to go ahead and actually set up a prediction for us here. So I'll select the Create a Prediction icon. And I'm going to go ahead and select this file with my amino acid sequence. I'm currently working with a FASTA file here. So I'm going to open that up. And after doing so, I'll scroll to the bottom of the screen, and I can see that particular file has become an unsubmitted prediction in my prediction list. The yellow icon you see over here to the left of the name indicates it as an unsubmitted prediction. I'll then go ahead and select this prediction, and then select the Run Prediction icon up here at the top. Now here, what NovaFold is doing is checking to ensure that your protein sequence fits within our 400 residue limit. While we're currently limiting our structure predictions to sequences 400 residues or less, we will be expanding soon as we optimize our algorithm for these larger structures. Now, NovaFold doesn't offer ahead of time the exact time that the cloud will take to process each prediction. However, we can estimate how long your prediction will take to run based on the number of residues in your sequence. So I'm going to select this estimated cloud time link here. And so here we're looking at the NovaFold benchmarks page on the DNASTAR website. So for small proteins, about 100 residues or less, the NovaFold prediction will only take a matter of hours. For larger proteins, which are 200 to 300 residues in length, the results may take a day or two to return. Now, if we look at this graph where NovaFold recreated the CASP-10 competition results, we can see that the results from this evaluation really do confirm those time estimates here. So I'm going to close this and go back to Protein 3D. We'll click Run. And after a couple seconds here, we'll see this protein sequence start to run. And yep, there it goes. We can see that once you see this green triangle icon here, this indicates that the prediction is running and you've started to use your cloud resources here. While this prediction is running, I can choose to safely close the NovaFold panel or Protein 3D and not impact this prediction at all since this is running up on the cloud. However, I will have to log back into Protein 3D to check if this prediction is complete to obtain my final results. So I'm going to go ahead and stop this prediction as I've already done some predictions for today's demonstration. Yep, so once I've stopped it, I can see this icon here that indicates that this prediction is terminated. So we can see from some of these completed predictions that there are really a variety of final run times here. Some take as little as 20 minutes, some take as many as 8 hours or even more. And again, there's no way to tell exactly how long your prediction will take, but as we looked at before, there are some pretty good estimate approximations based on the size of your protein. So some of these really small proteins will take much less time to predict. So once our prediction is complete, you can select it from this prediction list and view the results. So I'm going to open up this one EFU protein here and expand up our details panel, because our details panel over here on the side is going to be our most important panel for analyzing these particular predictions. So we can see over here in the details panel we have several different tabs with a lot of different information related to our prediction. First tab I want to point out is our Templates tab that I'll expand here. And this identifies the top 10 templates used in the threading approach discussed earlier. This tab displays the template and links to the PDB website. So if we click on any of these templates, we can pull up that particular entry in the PDB database for that particular structure. So we'll go back to NovaFold here. And in looking back at these templates, we can also see the coverage of that particular template to the query sequence, as well as the specific threading algorithm that was used to select that template here. I'm going to close down this Templates tab so we can look here at our Results panel. And our Results tab here displays the thumbnail images of our various predicted models. Now each model has a C-score, TM score, and RMSD. The CM score, the confidence score, ranges from -5 to 2, with 2 being the best possible. The TM score ranges from 0 to 1, with 1 being the best. And finally, the closer the RMSD, or the root mean squared deviation, is to 0, the better. These models are then color-coded green, orange, or red for their predicted quality, with the coloring based on the model's TM score. Green models are the highest quality predicted models and are appropriate for drug design, drug screening, ligand docking, and molecular replacement. I'm going to go ahead and open up one of these green models here so we can look at that displayed nicely up in this structure view. If you have some orange models, these may be of a slightly lesser model quality here. These are more appropriate for mutagenesis design and predicting active sites, disease substitutions, alternative splicing patterns, and domain boundaries. And finally, the red models may be useful for predicting domain boundaries. To be a green model, the TM score threshold needs to be greater than 0.75. To be an orange model, the TM score needs to be between 0.5 and 0.75, and red models fall below this 0.5 threshold. So as I've done before, clicking on the thumbnail of our model of interest will bring it up into the Protein 3D structure view for further analysis. And again, we can do all the same things that we did earlier within Protein 3D within this model, rotating it, applying different methods and analysis tools to it, as well as looking at the structure within the structure view. What's nice about the structure view within NovaFold is that it does have this nice color-coded line that corresponds to the colors in the structure resulting from your prediction. So with that, I'll take any questions that people have so far in the webinar.
Great, thanks, Jackie. We did have a couple of questions. One attendee writes, I'm an academic and I can use iTaser for free. Why would I use NovaFold?
Sure. So academics are able to use iTaser for free. What they don't get with NovaFold, or what they don't get with iTaser that they do get with NovaFold, as I discussed earlier, they do get that nice user-friendly interface and analysis methods that we have here. They don't have a wait time to get submitted into the queue. They also have the ability to run multiple structure predictions at once, and you have the full team of support staff and protein scientists here at DNASTAR that you can call or email or chat with to get any of your questions answered while you're working through submitting and analyzing your various protein predictions.
Thank you. And talking about the support that comes with NovaFold, can you talk a little bit more about what's included with that?
Sure, yeah. Actually, I think the best way for me to do that is to pull up our website here. I'll just go to the home page of our website. And all of our support options are here under the support tab on the website. As usual, as with traditional support, we do have the option to email, call. You can submit support requests and even chat with some sales representatives on the website. Additionally, we also have a lot of support options that you can take advantage of, such as videos. We have over 200 videos here, along with many videos specific to Protein 3D and NovaFold that you can take advantage of. And finally, our webinars that you're looking at today. So we have many different webinars on a lot of different topics, many of those on protein as well. So like I say, we have not only online resources, additionally help and different tutorials as well, but people who are happy to pick up the phone and help you work through your project.
Great, thank you. And what are my options for exporting images for publication?
Sure. What you can use here in NovaFold or Protein 3D actually, so you can set up your protein any way you would like within your structure view, set up coloring, rotations, surfaces, as I'll show you later in this webinar. You can really easily export those by selecting File, Export Image. And here you can export that image as high-quality PNG images, JPEGs, or GIFs. So you can use any of those exported images straight into your publication. Those give you nice, crisp, clear publication quality graphics for your use.
Great, thank you. And I think we have time for just one more question. We have one question here. What file formats are compatible with Protein 3D and NovaFold?
Yeah, so when you're going here, creating a prediction, or even bringing some sequences into Protein 3D if you're just looking at the sequences view. File formats that are supported are anything that you can download from PDB, so anything you bring in from the PDB website. Additionally, we also support FASTA files, as I brought in earlier, as well as GenBank files as well. Additionally, our Lasergene applications are integrated, so if you're already a Lasergene user and you have some sequence files saved in other Lasergene formats like SeqBuilder, Megaline, or Protean, you can bring any of those sequences into this application as well. Great. All right, I'll go ahead and move on here, showing you a couple other examples. So by housing NovaFold within Protean 3D, as we've seen here today, you do have the opportunity to take advantage of Protean 3D's graphical views and customization options that we discussed earlier in this presentation. I'm going to go ahead and close down. We're not going to use the NovaFold prediction I just showed you, but keep in mind that you can use any of these prediction results to do any of these following workflows that I'll show you. But for now, I'm just going to go ahead and use this example 7TIM structure again to walk you through this particular problem. So in the methods panel here, one of the most interesting methods that we've recently added is our DNASTAR Epitope prediction method. So what I'm going to do here is quickly walk you through how to use this particular prediction method to view the solvent-accessible surface of the likely epitope in relation to the rest of the protein. So in looking at the structure view, I first want to add a surface to this. And the way we're going to do that, we need to look at the Surfaces panel, which I'll open up by selecting View, Explorer, and then Surfaces. And this opens up this nice little Surfaces panel here in our Explorer. From the Surfaces panel, I'm going to want to go ahead and create a surface. So I'll select from the drop-down menu a solvent-accessible surface of high quality. So this selects the whole protein and adds this nice surface here, as we can see. But for this particular example, I do want to add a specific color to my surface. So the way I can do that is by going back to our Style panel that we've looked at earlier. So I'll open the Style panel, open the Color tab, and then choose to change our color for our current surface. So I'll change that to a green color here and click OK. So we can see that this entire surface then, we've added that green color to it. So now I'm going to go ahead and start the epitope prediction part of this particular workflow. So what I'm going to go do here is open up the analysis view. We'll take a break from looking at the structure view for a second. And then we want to add a new analysis method to this view. So I'll also open up my Methods tab, and then I want to check the box for B-cell epitopes. Now I'll scroll down to the bottom of the view here, and we can see that this B-cell epitope prediction method was added to my analysis view. And from this analysis view, we can expand this out and see some of these nice antigenic regions, which are the light pink, and highly antigenic models, which are the dark pink that are displayed. So I can also expand these subunits. So we have the 7TIM-A subunit and the 7TIM-B subunit. And I can expand those out and see some graphs for confidence scores for these particular regions. And these graphs here highlight the possibility that the region that we're looking at is a B-cell epitope. So if we have an region of interest, let's say I'm interested in this region here, I can select that. And we'll want to be able to view this particular region on the structure view. So after I've highlighted this region of interest, this highly antigenic region, I'm going to go back to the Style panel here, and I'll change the color. So let's say I want to change it to this kind of salmon color here, and click OK. So now if I go back to the structure view, I can see that we are now able to visualize this highly antigenic region within the context of the entire structure and the surface itself. So that gives you some really nice visualization tools where you can really closely observe that region of interest. So that wraps up that particular workflow. One of the other workflows that's really helpful to know in Protean 3D is how to open and align multiple structures within a single Protean 3 document. This particular workflow will allow you to discover similarities and differences between the two structures. So I'm going to go ahead and close out of this, and I'll open up a whole new project. So I've already downloaded two structures of interest from PDB that I want to align. And so I want to open them up both together here within the structure view in the Protean 3D interface. Now to do so, I will select File, Open Group, and then I will go ahead and keep this default setting of Center on Screen to allow similar structures since I want to eventually align these. I'll now click the Add button and select my two structures that I would like to import into the structure view. And then I'll go ahead and click OK. And now we can see here that the two structures are both open in a single document. But these are kind of a big jumbled mess right now, so we'll start going through this workflow to make them easier to analyze for our particular alignment. So the first thing I want to do to be able to look at this a little better is go to our Style panel that I have open here. And what we're going to do is choose to Color by Chain, which I'll select from this drop-down menu here. So after doing so, we can see that our 1BFD structure that I opened consists of a single green chain, while our 1PVD structure consists of both a blue and a yellow chain. So right now these chains are unaligned, so they just overlap with each other randomly on the screen. To align these particular chains, we'll first go to the Explore menu, and we'll select the chains that we want to align from the Molecules tab here. So I want to open up this menu a little more so we can work with it a little more easily. There we go. So now we can see all of the particular chains that we have open up here within this structure. So the ones I want to align are the A chain from 1BFD and the B chain from 1PVD. And so I'll select both of these by control clicking or command clicking. So now we want to align these selections by selecting Structure, Align Structures, and then I'll pick this rigid body alignment for the selected region, so this TM Align option. And we'll give that a second here. And our alignment is complete. So now that this is complete here, we can really easily, as before, zoom in and out of the structure if we want, or apply any of these other analysis methods. What I actually really want to do here is open up our Details panel. And so right now I'm looking at that one particular residue, but if I click off of that, there we go. We can really easily look at this RMSD value, which is what we're most interested in after the particular alignment, to determine the degree of similarity between these two proteins. So as discussed with our NovaFold structure predictions, the closer our RMSD value is to 0, the better. So an RMSD value of less than 0.5 indicates near identical structural similarity. 0.5 to 0.2 or 0.5 to 2 indicates still a very high degree of similarity, and an RMSD of 2 to 5 indicates a lower but still significant and noticeable degree of similarity. So from previous research on these two protein structures, I already know that these two structures have, or these two sequences have a 19% sequence similarity. And so this corresponds really well with our RMSD value of 3.342 that we got out of this particular alignment.